ladies and gentlemen. First, I would like to thank CECOG for this invitation. It's a pleasure to be part of an in-person event after 1 year of virtual meetings and webinars. And I'm lucky to get this interesting title, Targeting KRAS Mutations. I would extend the title to KRAS, the undruggable target, because after years and years of research, we have still no, up to now at least, no effective treatment in the clinic. And why this is important, you can see here on this slide. This is the pie charts which are necessary for lung cancer, showing the oncogenic drivers in lung adenocarcinomas. The first thing you can see here is that we have two charts. One on the left side is in early stage patients, and on the right side for metastatic patients. And the good thing is they are pretty similar. That means that one can speculate that the results we get in the metastatic setting can be at one time translated to the adjuvant setting, which may lead then actually to cure of patients with these types of lung cancer. What you can also see here that there are two big pies, big slices of these pies, namely the KRAS and the EGFR piece. KRAS stands for Kirsten-Rast sarcoma gene, EGFR for epidermal growth factor receptor, and as you can see, these are the genes which are mainly affected in lung adenocarcinomas. 30% KRAS mutations and approximately 30% EGFR mutations are present in these patients. What you can also see here that we have a lot of other aberrations which are pretty rare in some cases. For example, here RIT is present only in 0.2% of the patients, and one important marker, NTRK, is not part of this pie, which is unfortunate because we have effective treatments available in these patients, which is the case also for all the other targets or many other targets. That means that we have to search for these mutations. We have to do comprehensive genomic profiling of these patients in order to find these rare events and to treat patients effectively. So in Austria, we are in a lucky situation that we have this comprehensive testing, and we have also many drugs available also in the clinics. They are approved already. For example, for EGFR, of course you know that for ALK fusions, for ROS1 fusions, RET fusion, HER2, and so on. So these treatments are available in our patients, which is really a good sign because they are really effective. And this is different to other countries in the European Union and also in other places in the world where these drugs are not available. So this is another pie chart specifying now the KRAS piece. And as you can see here on the second circle here, these are representing the KRAS mutations. So they are specific in two codons, codon 12 and codon 13. In lung cancer, we have one main mutation is the G12C mutation. That means that glycine is substituted by cysteine, and this is present in 40 to 50% of all KRAS mutation. That's why it's very important. Another important thing is, if you look at the two main mutations, EGFR and KRAS, that EGFR mutations are mainly present in never smokers, so 42% of never smokers harbor this EGFR mutation, whereas in former smokers and current smokers, KRAS is the main mutation. 30 and 35% of these patients have the KRAS mutation, and very low numbers, 5% and 13% of EGFR mutations. Why is this important? Because never smokers are expected to have better genomic profiles compared to patients who smoke. There are more aberrations, more mutations, and maybe they are more difficult to treat despite of this fact. So KRAS mutations are more often present in smoking people. The distribution of KRAS mutations is also different in the various solid tumor types. Here we have the example of pancreatic cancer and colon cancer and lung adenocarcinomas. And as you can see here, in pancreatic cancer and in colorectal cancer, the main mutation is the G12D mutation. So glycine is substituted by an aspartic acid, whereas, as I mentioned before, in lung adenocarcinomas, it's a G12C mutation, glycine to cysteine. Why is this important? Or just first, a look into the mechanism of KRAS. So KRAS is a membrane-bound GTPase. It's inactive if it's bound to GDP, to guanosine diphosphate. This complex is activated by guanosine nucleotide affecting factors such as SOS1 or SHP2. They phosphorylate this GTP to GTP, guanosine triphosphate, and this is the active form of KRAS, which then can activate the downstream signaling pathways, the MAP kinase pathway and the PI3 kinase pathway, for example. The circle is then finalized by the inactivation of RAS by gap proteins, NF1 for example, which inactivate KRAS and bring it back to the GDP status. This circle is interrupted if KRAS is mutated, here in position 12, for example. In this case, KRAS is always in the active form, and all the downstream pathways are constitutively activated, and that means that oncogenic signaling takes place. Now, with the new inhibitors, which directly affect this mutation at codon 12, for example, sotorasib or adagrasib, which are two of the inhibitors which has recently been granted by FDA as a breakthrough therapy designation. So they should be important. And as I said, this circle is interrupted by these inhibitors. KRAS cannot activate the downstream signaling pathways, and the circle is interrupted. So in the past, indirect KRAS inhibition was studied, for example, blocking of KRAS expression or inhibition of KRAS processing or inhibition of upstream or downstream signaling, but also by specific inhibitors of RAF, MEK, ERK, and PI3 kinase pathway inhibitors. Now the strategy has changed because of new information on the structure of KRAS. And direct KRAS inhibitors have been developed, such as covalent KRAS G12C inhibitors or pan-KRAS inhibitors, which also affect other mutations other than the G12C mutation. So this is just a list of historical attempts to target KRAS in non-small cell lung cancer. All of these trials failed. I want just to point out one specific inhibitor, selumetinib, which is a MEK inhibitor, which has extremely promising phase 2 results. In this trial, selumetinib plus docetaxel was more active than docetaxel alone. High response rates, 37% objective responses, and also a doubling of the PFS time from 2 months to over 5 months. So this was really exciting. But then they performed the phase 3 trial, and this was a negative trial, so there was no difference in efficacy between the selumetinib combination and the chemotherapy alone. So that's why I mentioned KRAS as undruggable target. But fortunately, as I said, structural, the structure of KRAS was studied in more detail, and they found a region in KRAS called the switch 2 region, which is pretty close to the GTP binding site, and this is a pocket where the chemists could design drugs, small molecules, which fit in this pocket and block KRAS activity. And you can see the importance of this target, because many companies developed inhibitors, specific inhibitors for this G12C region. And the two drugs which are ahead of the others are adagrasib and sotorasib, where we have also clinical trial results available. So this is just to show you where this drug binds to KRAS. So this is the switch 2 region I mentioned before, and the C12 position, the codon, the cysteine at codon 12, which shows you that this, in this case, sotorasib binds covalently to this cysteine at position 12. Which then interferes with the GDP pocket, which is pretty close to the switch 2. The difference from sotorasib to ARS and adagrasib are in this hydrogen bond here. So all the other two inhibitors bind covalently to this C12 position, but they also have this hydrogen bond with a cysteine at position 95, which means that this drug should be more specific and also have a high activity. In theory, we'll see what the clinical data will tell us. So sotorasib was studied in a phase 2 trial. The trial was called CodeBreak 100. The results were published recently in the New England Journal of Medicine, and in this paper, sotorasib, the efficacy of sotorasib was studied in patients with non-small cell lung cancer. In this phase 2 trial, 126 patients were included. Key eligibility criteria were locally advanced or metastatic non-small cell lung cancer. This KRAS G12C mutation must be present, assessed by central testing. Patients must have progressed on prior standard treatments, and no brain mets were allowed. So sotorasib was orally administered at 960 milligram once daily until disease progression. This is pretty interesting because today there was in the issue of JCO, it was published today, this was discussed because at this dose there were, let's say, some side effects which might not be present at a dose of 240 milligrams, which was also discussed. And therefore, FDA and some investigators had the opinion that 240 milligrams is a better dose because it should be equally effective but had less side effects. And so FDA forced or asked Amgen to study this dose together with 960 to find out what is really the best dose for the patients. If the efficacy is similar and the side effects are reduced, of course, the lower dose would be better for the patients. So Amgen has time until February 23 to show FDA the results. But coming back to this trial, the primary endpoint of this trial was objective response rate, blinded by independent central review. The key secondary endpoints were disease control rate, progression-free survival, overall survival, safety, and they had some exploratory endpoints as evaluation of biomarkers such as PD-L1 or co-occurring mutations. So this is the response to this sotorasib treatment by central independent review. Objective response rate 37%. which is interesting because it's exactly the same objective response rate as in the selumetinib trial, 37%. Disease control rate 80%. We have just a few complete responses, 34% partial responses, but mainly stable diseases, 44%. The median duration of objective response was 11 months, which is it's quite okay. So this is the waterfall plot. Again, what you can see here is that there are many stable diseases, some partial responses, and just a few complete responses. Just for comparison, this is the waterfall plot of adagrasib, the other compound. Unfortunately, we have only the response data available. We have no data for PFS so far. But in these 51 evaluable patients, the objective response rate was 45% and disease control rate 96%, so a little bit better than with sotorasib, but comparable. This is the PFS data for sotorasib. Median PFS is 6.8 months, which is quite good for this patient population, where I expect a median PFS of 2 months, at least with docetaxel treatment. So a tripling of PFS time, at least in this phase 1/2 trial. We will see whether this will hold true also in the confirming phase 3 trial. Median overall survival is 12.5 months. As I said, for this population, it's pretty good. With regard to the exploratory biomarker analysis, they have 2 studies. First, the comparison with PD-L1 expression, which nicely shows here that the response is decreasing with higher levels of PD-L1. So the best response rates have patients with low PD-L1 expression, so less than 1% PD-L1 means that the response rate is 48%. For the group with 1 to 49% PD-L1 expression, the response rate is 39%, and the lowest response rate, 22, was achieved in patients with equal or higher than 50% PD-L1 expression, which is pretty interesting because this would mean that this inhibitor is more effective in patients with low PD-L1 expression, so this should be the primary target population. Or you can combine this drug with checkpoint inhibitors to improve the outcome. At least it could be a possibility. The other study was comparing the response with co-occurring mutations such as p53, STK11, and KEAP1. For p53 and STK11, there was no difference between wild type and mutated protein. But for KEAP1, if KEAP1 is co-mutated with KRAS, then these patients have a lower response to sotorasib. What they also did was analysis of plasma circulating tumor DNA, which is important also maybe for diagnostics, at least in patients where tissue results are not available. Maybe they can use plasma for the confirmation of the mutation. And they were able to collect blood from all 126 patients of this phase 2 trial. They tested all the patients with Guardian 360, which is an NGS method. And this was possible in 112 patients with positive results in 78 patients. Negative results were achieved in 31 patients, and in 3 patients the testing failed. So they combined these patients from the trial with additional patients, just to have more patients for the comparison between tissue and plasma, and they ended up with 189 patients. The good thing is the response rate between patients who were selected by tissue were similar to patients who were selected by plasma. So as you can see here, the complete response rate is low in both cases. Partial responses were present in 35%, 36%, stable diseases in 43.5 and 41.6%. Also the progressive disease is similar in both groups, and also the missing patients. So the objective response rate was 37% in the case of tissue selection and 36% in case of plasma selection. Also, this control rate is pretty similar. With regard to the concordance between tissue and plasma, the overall agreement is pretty good with 81%. Also the negative agreement, which is 100%, is also pretty good. The positive agreement is a little bit lower, but is still okay for this type of analysis. So I think it's the same situation as in the EGFR field, and I think this plasma assays can be used in addition to the tissue in selected patients. So tissue is still the gold standard, but if no tissue is available, I think you can also try to get the result from the plasma. If it's positive, then it's good for the patient because you can deliver the drug. If it's negative, then it might be also false negative. That should you keep in mind at least. So we have this exciting phase 2 results, and so it's important to confirm these results in a phase 3 trial, not to end up as in the selumetinib story that the phase 2 trial was exciting, but the phase 3 trial failed, and showed this significant difference. For this trial, which is called CodeBreak 200. It's a phase 3 trial for advanced or metastatic patients with KRAS G12C mutations. The key inclusion criteria were that patients should at least progress on at least one prior systemic therapy with chemotherapy and a checkpoint inhibitor, good performance status, no brain mets, and no treatment with docetaxel. These patients were randomized to sotorasib, 960 milligram oral daily, and docetaxel until disease progression, and also long-term follow-up will be collected. The primary endpoint of this trial is progression-free survival. Secondary endpoints are overall survival, objective response rate, duration of response, time to recurrence, disease control rate, and safety and tolerability. So I think we all have to wait for this trial in order to can safely say that we will really have an active treatment for this type of disease. So what are the questions for the future development of KRAS G12C inhibitors in non-small cell lung cancer? Will it still be monotherapy, or will it be more effective with combination strategies? So when you saw the efficacy of the monotherapy, I doubt that this will be the way how it should be moved forward. So I think combination strategies will be more effective than the monotherapy alone. Is there a role of KRAS G12C inhibitors in the first-line setting? Well, as monotherapy will be questionable, but maybe as in combination with other drugs, it could be also translated into the first-line setting. And one question could also be, what are the mechanisms of resistance? So combination strategies for these inhibitors could be, for example, a combination with EGFR TKIs, which will be studied in the CodeBreak 101 trial. Combination with immune checkpoint inhibitors, which is also studied in several trials. Combination with SHP2 and SOS1 inhibitors, or a combination with MEK inhibitors might be also interesting and will be studied also in the CodeBreak 101 trial. So this is a summary of all the combinations I mentioned before. Of course, combination with EGFR TKIs would be very interesting. SHP1, SHP2 and SOS1. Of course, all the combinations with the downstream factors, MEP kinase inhibitors, MEK inhibitors, or mTOR inhibitors, and also a combination with immune checkpoint inhibitors could be successful. So what are the potential mechanisms of resistance to these inhibitors? Of course, as in other targeted drugs, secondary mutations in the drug binding pocket or oncogenic hotspot mutations, gain of function mutations in MEP kinase pathway members possible, for example, mutations in NRAS, BRAF, MEP kinase, RETRIT, or EGFR. Then loss of function mutation in tumor suppressors such as NF1 or PTEN, oncogenic fusions, such as RET fusions, ALK fusions, or fusions with BRAF, and of course gene amplifications in KRAS, MET, or EGFR might also be potential resistance mechanisms. So I'm coming to my summary. So KRAS is one of the most, or is the most frequently mutated oncogene in non-small cell lung cancer. That's why small molecule inhibitors that target specifically these KRAS G12C mutations were developed. The first clinical trials with sotorasib and adagrasib showed promising activity in non-small cell lung cancer patients. That's why FDA has granted breakthrough therapy designation to both inhibitors. Several resistance mechanisms were identified, and of course, molecular testing or comprehensive genomic profiling is important at diagnosis. Thank you for your attention.
Thank you, Martin, very much. I apologize that it's to sneak out for a moment, but we have 6,000 infections as of today, and everybody's very nervous within the country and the county. So this was a call I could not deny. I apologize for that.
That's not good news.
I apologize for that. So the question is here, which also was asked on the internet a couple of times, are we not too specific by targeting just one mutation? Because when you showed the different mutations that are present, the question came up whether it would not be wise to look for a broader spectrum of tyrosine kinase inhibition.
Yeah, that's a good question. So one argument for these specific inhibitors is that 50% of all KRAS mutations are, at least in lung cancer, are this G12C mutation. So that's why I think the interest is higher in a specific mutation, because it's the most frequent KRAS mutation in lung cancer. But on the other hand, of course, so-called pan-KRAS inhibitors are developed, and they will also target the other mutations. So we will see whether this strategy is more effective than the specific strategy. But I think, as you can see on the results, which are exciting for this population with a poor prognosis. And if you compare it with the 2 months median PFS with docetaxel, they showed pretty good results with a tripling of PFS time, so it should be kept in mind. But yes, this is one of the concerns, that it might be too specific, but we will see how it competes with the pan-KRAS inhibitors. But I also pointed out that KRAS monotherapy will or may not be the way how the drug should be moved forward, because this is a mutation of smokers. That means that these tumors, let's say, have a worse genomic profile compared to never smokers or non-smokers. That means that maybe one specific drug is not effective enough. So a combination of a smart combination with other drugs might enhance efficacy, and I think this will be the way how this drug will be moved forward.
This goes very well with the next question, which I just read from here, and that is, thank you very much for this excellent overview. which supports the idea that this was the drug of the year or the development of the year. Are we afraid of combinations of checkpoint inhibition plus KRAS inhibition because of toxicity, obviously?
That's a good question. I think we have to test this in clinical trials and see how the safety profile looks like. But I think from the theory, this could be a good combination, at least in patients with a high PD-L1 expression, as the trial results showed us that the drug is less effective in patients with high PD-L1 expression, which are more likely to respond to the checkpoint inhibition. So I think it would be a good strategy to combine both drugs, and we will see how the safety profile look like.
Thank you very much. So I'm through here with the questions which came in via the chat, the internet chat. I'm turning now to the auditorium. Do you have any questions, ladies and gentlemen? Maybe we could ask one thing, which is, of course, very worrisome. I mean, obviously, this is very specific, G12C, right? So G12D is out of the game, right? Because that's what everybody was looking at with pancreatic cancer. This is something where you would have a certain inclination to. It does not work at all, right?
Well, this is more effective for the G12C mutation. I have no data for the other mutations, but because it's specifically developed for this mutation, I think it's specific, and the efficacy is higher in these patients. Whether it affects also other mutations, I think, at least I don't know the data for this. But I'm sure that other inhibitors specifically targeting the other mutation, namely the G12D mutation,
Right.
which is present in pancreatic cancer and colorectal cancer, will be developed, and or the pan inhibitors, the pan KRAS inhibitors, which might be also pretty interesting in all solid tumors.